Good evening, everyone. Hello, students. How are you all? So, poll has been launched. Quickly answer through poll. My audio is clear and my screen is visible. So, today we are going to discuss some of the questions which came in test. So, today's lecture will be a paper discussion class. Let's start with the question number 1, which says select the wrong statement. Option A is replication of DNA takes place in S phase. B is recombinase enzyme initiate crossing over in Packetine stage. C is removal of chiasmata is called terminalization of in dikinases. D is dissolving of synaptonymal complex occurs in anaphase 1. What do you think? Which one is the wrong statement? Try to recall the stages of prophase 1, of meiosis 1. We know that the prophase 1 of meiosis 1 is very long and complex because this consists of 5 stages. Number 1 is a lactotine, where the chromosome they become gradually visible. Next stage is the zygotine stage. In this stage, the pairing occurs between the homologous chromosome and is known as synapses, and the complex form is known as a synaptonymal complex. Very good, Shishant. And the pairing between the homologous chromosome is known as synapsin. And the complex form is known as synaptonymal complex. After zygotine is the Packetine stage, where crossing over occurs. And the crossing over takes place between the non-sister chromatid of the homologous chromosome. Remember this. Here the enzyme involved is recombinase. And the recombination nodule are formed. After Packetine is diploatine. And in diploatine, dissolution of synaptonymal complex occur. But when the synaptonymal complex dissolve, but the chromosome, they remain attached at the site where the crossing over has taken place. So they form a X-shaped like structure, which is known as chiasmata. Last stage is dikinases. In this stage, terminalization of chiasmata occurs and the chromosome, they get separated. So these are the 5 stages of prophase 1 of meiosis 1. Now, look at the option. One says the replication of DNA takes place in S phase. Yes. Second is the recombinase enzyme initiate the crossing over in Pachytene stage. Yes, because the Pachytene stage is known for recombination or the crossing over between the non-sister chromatid of the homologous chromosome. C is removal of chiasmata is called terminalization in dikinases. Yes. Dissolution of chiasmata, it occurs, or the terminalization of chiasmata, it occurs in dikinases. Dissolving of synaptonemal complex occur in anaphase 1. This is a wrong statement. Dissolving of synaptonemal complex, it occurs in the diplotin stage. Remember this. So the correct option with respect to the question is D. Remember all the events which are taking place in the respective stages of the prophase 1 of meiosis 1. These are important, okay? Remember this. Next one is hnRNA undergoes now additional process. Out of them, in one process, an unusual nucleotide, which is 7-methylguanosine, is added to the 5' end of a molecule. What would you call this? Option A is tailing, option B is splicing, option C is termination, option D is capping. What do you think? What will be the correct option? Very good, Ritu. They are talking about the post-transcriptional events. And post-transcriptional event, it occurs in eukaryote. Remember this. And these post-transcriptional event, it includes capping, tailing, and splicing. Capping occur at the 5' end of the mRNA. Tailing occur at the 3' end. And splicing is the removal of introns. This capping and tailing is done to protect the mRNA from the exonucleases which are present in the cytoplasm, because in eukaryote, you know that the transcription takes place in nucleus, right? And this mRNA, it moves from the nucleus to the cytoplasm for translation, because ribosome, they are present in the cytoplasm. And the capping is also important for translation also, because the ribosome, they recognize this capping which is present at the 5' end. And also remember one point, that at 5' end, 7-methylguanosine cap is added. And the enzyme involved is guanine 7-methyltransferase. I'm giving you important points, okay? The question is asking that the hnRNA undergoes now additional process. One of them, out of them in one process, is an unusual nucleotide, which is 7-methylguanosine, is added at the 5' end of the molecule. So 5' pe capping is taking place. So the correct option is capping. I hope it is clear to everyone. Next is question number 3 says, which of the following feature is associated with the trisomy of 21st chromosome? One is flat round face, Second is mental retardation. Third is presence of gynecomastia. Fourth is loss of skin pigmentation. What do you think? What will be the correct option? They're talking about the trisomy of 21st chromosome. That means they're talking about Down syndrome. Try to recall all the symptoms of Down syndrome. Then you can easily answer this question. Very good, Ritu. In Down syndrome, the symptoms, number one is small round head. Flat back of head. Short height. Furrowed tongue. Broad palm. Partially open mouth. Mental retardation. Remember all the symptoms, okay? Now let's look at the option. It says the number one says the flat round face. Yes. Second said mental retardation. Yes. Third says presence of gynecomastia. Presence of gynecomastia is the symptoms you see in Klinefelter syndrome. Where you will observe an extra copy of X chromosome. And this Klinefelter syndrome is mostly observed in males. Loss of skin pigmentation, no. So there's only one and second statement which is related to the Down syndrome. So the correct option is B, which is only 1 and 2 are correct. I hope it is clear to everyone. Next, which is the correct link reaction? Student, remember that it is dehydrogenase. Okay, it's a misprint. What do you think? What will be the correct option? Which is the correct link reaction? You know, in glycolysis, glucose is converted to pyruvate. Right? This pyruvate enter into mitochondria, where this pyruvate is converted into acetyl-CoA. And this reaction is catalyzed by an enzyme which is known as pyruvate dehydrogenase. And it's an oxidative decarboxylation reaction. Remember this. Decarboxylation means like CO2 is released. Here you can see the CO2 is releasing. And this acetyl-CoA then enters into the Krebs cycle. And in the Krebs cycle, NADH and FADH2 are formed. ATP and CO2 are also formed, okay? But here I'm talking about the NADH and FA2H2. This NADH and FADH2, they will enter into the electron transport chain, which occur in the inner membrane of the mitochondria. And finally, you get the ATP. Okay? Now let's look at the options. So when this pyruvate is converted into acetyl-CoA, CO2 is released. NADH dot H plus is formed. That means here NAD plus would be the reactant, and here coenzyme E. Now you can easily find which is the correct link reaction. Pyruvic acid, NAD plus, coenzyme A, enzyme is pyruvate dehydrogenase, acetyl-CoA is formed, NADH plus is formed, and CO2 is released. In the second one, there's involvement of FAD plus. This is not the correct link reaction. In C option, there's NADP plus. This is also not the correct link reaction. And in the D, you can see it's an oxidative decarboxylation reaction. That means CO2 has to release. There's no CO2 in option number D. So the correct option or the correct link reaction is option number 1. I hope it is clear to everyone. Okay. Now move to the next one. Which one of the following statement is correct? Sporogenous tissue is haploid. Endothecium produces the microspore. C is tapetum nourishes the developing pollen. D is hard outer layer of pollen is called intine. What do you think? What will be the correct statement? Very good, Veena. Very good, Sushant. You know, in a typical anther, there are 4 layers, right? Outer is the epidermis. After epidermis, there's endothecium. And this endothecium, it has radial thickening. Inner to the endothecium are middle layers. And then tapetum. First 3, epidermis, endothecium, middle layer, they all perform function which is protection. They also help in the dehiscence of the anther. When the anther get mature, they help in the dehiscence of the anther. So that the pollen, they get released out. Tapetum, its function is nutritive in nature. It provides nourishment. To the developing pollen. And inner to the tapetum, the sporogenous tissue are present. And each cell of the sporogenous tissue, they form microspore mother cell. These microspore mother cell, they undergo meiosis and they produce microspores. These microspore, on maturation, they form a pollen grain. And the pollen grain, they have two layers. Outer is exine, inner is intine. Exine is hard and it is consists of sporopollenin. And intine is thin and it is consists of cellulose and pectin. Now look at the option. A say the sporogenous tissue is haploid. No, it is diploid. That is why the meiosis is taking place and the 4 haploid microspores are forming. B is endothecium produces the microspores. No. Microspores are forming from the microspore mother cell, which is undergoing the meiosis and forming the microspores. And the process is known as microsporogenesis. Genesis means formation and microspore, because here the formation of microspores is taking place, so we call it a microsporogenesis. Third says the tapetum nourishes the developing pollen. Yes. D says the hard outer layer of pollen is called intine. No, it is exine. I hope it is clear to everyone. So the correct option is C, which is tapetum nourishes the developing pollen. Next, choose the correct combination for given flow chart. What do you think? What will be the correct option or the correct sequence? Okay, now let's discuss. Very good, Sushant. See, this is a nucleus. And always remember, the outer membrane of the nucleus is continuous with the ER. Okay, remember this point. Here it is given rough endoplasmic reticulum. So I'm drawing ribosome here. This is a rough endoplasmic reticulum. And we know that function of the rough endoplasmic reticulum is protein synthesis because it contains ribosomes and ribosomes are involved in protein synthesis. Let's say is a vesicle which bursts out and it contains the protein. This vesicle is the transport vesicle. Because function is to only transport the material. So it's transporting the material from the ER to the Golgi. This is the cis phase of the Golgi. These are the medial cisterna. This one is the trans phase or the mature phase. Cis phase is also known as the forming phase. So this transport vesicles, which are containing the protein, they will fused with the cis phase of the Golgi. And the function of the Golgi is in the modification of the protein or the lipids which are received from the ER. Okay. After modification, the vesicle will bud out from the trans phase of the Golgi and it containing the modified proteins. This is the secretory vesicle. Because the function of this vesicle is to store and secrete the material. Now this vesicle is the plasma membrane. Now the vesicle which is bud out from the trans phase of the Golgi containing the modified protein, which is the secretory vesicle. Now this vesicle will fuse with the plasma membrane. And all the material which are present in this secretory vesicle, they will release out. This is known as exocytosis. Exo means outside. Okay. Now look at the options. A is the transport vesicle. From the trans Golgi, the secretory vesicles are formed. These secretory vesicles then fuse with the plasma membrane. So A is transport vesicle, B is secretory vesicle, C is plasma membrane. So the correct option is C. Okay, I hope it is clear to everyone. Moving next, find out the correct match from the following table. What do you think? Which statements or the matching are correct? Now look at the question. They're asking, one says that the primary CO2 acceptor in C3 plant is RUBP. Yes, the primary CO2 acceptor in C3 plant is RUBP. Ribulose 1,5 bisphosphate. In C4 plant is not RUBP, it is PEP, phosphoenol pyruvate. So first is incorrect. Second says the primary CO2 fixation product during Calvin cycle is PGA or 3-PGA. They are saying that primary CO2 fixation product, primary means first CO2 fixation product is 3-PGA. The first product we are getting in Calvin cycle is 3-PGA in case of C3 plant. And its C4 plant is also 3-PGA. See, in C4 plant, we know in C4 plant that they have Kranz anatomy. The Calvin cycle also takes place in C4 plants. Remember this very important point where many students they get confused. Okay, please remember that Calvin cycle also takes place in C4 plant and it occurs in bundle sheath cell. Okay, it occurs in bundle sheath cell. So here also in bundle sheath cell, the Calvin cycle is taking place and they're asking about the Calvin cycle. So here also the first CO2 fixation product during Calvin cycle will be 3-PGA. So second statement is correct. Third says the number of carbon in the primary CO2 acceptor. In C3 plants, we know that the RUBP is the primary CO2 acceptor, but it's not 3 carbon, it's 5 carbon. And in C4 plant, phosphoenol pyruvate is the primary CO2 acceptor and it is 3 carbon, but the RUBP is 5 carbon, so this is wrong. Fourth is the initial carboxylation reaction occurs. In C3 plant, the Calvin cycle occur in the mesophyll cell. And in C4 plant, they're asking about the initial carboxylation reaction. Here it is also taking mesophyll cell, and this is the carboxylation reaction in C4 plant. It is also taking place in the mesophyll cell. So fourth is also correct. Second is correct, fourth is correct. So correct option would be B, which is second and fourth. I hope it is clear to everyone. Moving next, match the column 1 with column 2 and choose the correct option. Pacific salmon fish, it breed only once in lifetime. NT equals to not ERT. This is an exponential growth. A second. P3. Oyster, a large number of small sized offspring. Fourth. And D, this is a logistic. D shows the logistic growth. So D1. Very good, Veena. A2, B3, C4, D1. Correct option is D. Moving next, find the incorrect match from the column. Icomycetes includes albugo, mucorhizopus. Ascomycetes includes claviceps, neurospora. Remember, the neurospora is used for biochemical and genetic work. Important, okay? Remember this. Aspergillus. The morels and truffles are also present. Morel and truffle, they are the edible fungi. Remember this. Basidiomycetes. Agaricus. Puccinia, which causes rust disease. Astilago causes smut. Deuteromycetes, it's an imperfect fungi. Remember this, because sexual stage is not known in Deuteromycetes. Remember this. It includes Alternaria. Coleotrichum. Trichoderma. Remember all the examples, okay? It's important. Option column 1 says the Astilago belong to class Basidiomycetes, commonly known as club fungi. Yes. Basidiomycetes, they are known as club fungi, mushrooms, they are also known as puff balls or bracket fungi. These are the alternative name of Basidiomycetes. Ascomycetes, they are known as sac fungi. Second says Agaricus belong to class Basidiomycetes, mushroom. True. Alternaria, sexual cycle is formed? No. Perfect, no. They are called imperfect. And Alternaria belong to Deuteromycetes, where and they are known as imperfect fungi because sexual stage is not known. Fourth says Aspergillus, sexual cycle is found called sac fungi. Yes, Aspergillus belong to Ascomycetes, and Ascomycetes, they are commonly known as sac fungi. So find the incorrect match from the column is 3. So correct option is C. I hope it is clear to everyone. If the pituitary gland of an adult human is surgically removed, which of the following endocrine gland would be less affected? Now, we know that pituitary gland, they produce a lot of hormones. Number 1 is that they produce growth hormone. They produce melanocyte stimulating hormone. They produce gonadotropins hormones, which are FSH and LH. They produce prolactin. They produce thyroid stimulating hormone. Oxytocin, vasopressin. Remember this, that oxytocin, vasopressin, they are synthesized in hypothalamus but released from the posterior part of the pituitary. And they also build ACTH, which is adrenocorticotrophic hormone. This adrenocorticotrophic hormone, it stimulates the adrenal cortex. To synthesize a steroid hormone, which is glucocorticoid. We know that adrenal gland, it has two parts, the adrenal cortex and the adrenal medulla. Adrenal cortex releases glucocorticoid hormone, which play a very important role in carbohydrate metabolism. It also produces mineral corticoid hormone. And adrenal medulla, It produces epinephrine, norepinephrine, which are the flight and fight hormones or the emergency hormone. So question is asking, if the pituitary gland of an adult human is surgically removed, which of the following endocrine gland will be less affected? If we remove the pituitary gland, obviously thyroid will be affected, gonad will be affected, and adrenal cortex will be affected, but adrenal medulla will not be affected or less affected. Okay, so the correct option is B, which is adrenal medulla. Moving next, the following diagram depicts the arrangement of different type of teeth in the upper jaw on one side and the socket on the other side. What is the correct option for the number of roots in incisor, canine, premolars, and molars of the upper jaw? What do you think? What will be the correct option? Incisor, they have one root. Canines, they have one root. Premolars, in upper jaw, they have two roots. In lower jaw, they have one root. Molars, in upper jaw, they have three roots. In lower, they have two roots. You can also see in the image, like sixth. Look at the sixth teeth. You can see 1, 2, 3. There are three roots. Here you can see in the fourth, 1, 2. There are two roots. Right? So they're asking about the upper jaw. Remember these roots, okay? It's important. Incisor 1, canine 1, premolar in the upper jaw 2, molar in the upper jaw 3. So the correct option would be B. Moving next, analyze the figure given below, the Km value for the curves of the four different pairs of enzyme and their substrates. PQRS are 1.72, 0.25, 3.40, and 5.75. So this is a Michaelis-Menten graph, okay? Michaelis-Menten shows that the velocity or the rate of the reaction, they are dependent on the substrate concentration. Okay, when the substrate is present at the very low concentration, so when we increase the concentration of the substrate, the rate of the reaction or the velocity of the reaction also increases. This is a point where the velocity is maximum. This x-axis shows the substrate, y-axis shows the velocity or the rate of the reaction. But it is up to a certain limit. Okay, because if after some time, if you keep on adding the substrate to the solution, the enzyme or the rate of the reaction, it won't increase because the enzyme, they are already saturated. And there's a Michaelis-Menten constant, which is known as Km. Km is the concentration of the substrate at which the reaction velocity become half of its maximum velocity. Can say maximum velocity is the Vmax. This is the Km shows the concentration of the substrate where the reaction velocity reaches half its maximum velocities. So we denote it by Vmax by 2. And remember one important point. Lower the Km. It means or it describes the enzyme has the greatest affinity for the substrate. Lower the Km means that at a very low concentration of the substrate, the reaction reaches half of its maximum velocity. Okay, so if the value of the Km would be low, that means the enzyme, they have a greatest affinity for the substrate. Higher the value of Km, that means the enzyme, they have lower affinity for the substrate or the reduced affinity for the substrate. Remember this point, okay? Important. Now P is given 1.72. Q is 0.25. R is 3.40 and S 5.75. So Q is having the lowest Km. That means the Q enzyme has the greatest affinity for the substrate. S is having the maximum Km. That means the S enzyme, it has the lowest affinity for the substrate. This is the correct order. Okay? Because they're asking the correct order about their enzyme substrate affinity. Q is having the maximum affinity for substrate. S is having the lowest affinity for the substrate. So the correct option is B, Q, P, R, S. Next is a person passes excessive urine and drinks much water, but his glucose level is normal. It is due to. It is due to the reduction in the vasopressin secretion. Vasopressin, the another or the alternative name is ADH, which is an antidiuretic hormone. Its function is to reabsorb the water and minerals or electrolyte. If vasopressin is produced in a very less amount, then the water reabsorption will be reduced. That means a large amount of water will be lost through urine. So the correct option is D. Moving next, match the excretory function of the section 1 with the part of the excretory system in section 2. Choose the correct combination from among the given answers. Malpighian corpuscle. It includes glomerulus plus Bowman capsule. And malfusion corpuscle ultrafiltration occur. Next come is PCT, where maximum absorption occur. Next is Hanley's loop, and Hanley's loop is consists of ascending limb and the descending limb. In the descending limb, water is reabsorbed. In the ascending limb, NaCl is reabsorbed. So in the Hanley loop, you will observe concentrated urine. Next one is DCT, where the reabsorption of water and NaCl occur. And finally is the collecting duct. Very good, Veena. So one in ultrafiltration is D. Concentration urine, loop of Hanley. Transport of urine, ureter. Storage of urine, urinary bladder. One D, second A, three B, fourth C. The correct option is A. Moving next, in the pelvic girdle of man, A, B, C, D, and E respectively represent. So pelvic girdle, it helps in joining the two hind limbs with the central axis of the body. So the pelvic girdle, it consists of two coxal bone. And each coxal bone is formed by the fusion of three bones. Which includes number one, ilium. Number two is ischium. Number third is pubis. And in ischium, there is a cavity is present which is known as acetabulum. And at this cavity, the thigh bone get articulated or joined. And these pelvic girdle or the coxal bones, they are joined ventrally by the pubic symphysis, which contains the fibrous cartilage. So A represent ilium, E represent pubis symphysis. B is acetabulum, the cavity. And remember, this cavity is present in ischium. D is ischium. C is pubis. A ilium, B acetabulum, C pubis, D ischium, E pubis symphysis. So the correct option is B. And this hole is known as obturator foramen. Now, moving to next question. Choose the correct option for A and B. Here you can see this nitrate is reduced to nitrogen gas. So here denitrification is occurring. And the enzyme involved in denitrification are thiobacillus and Pseudomonas. In this red color part, nitrification is showing where the ammonia is first converted to nitrate. Sorry, nitrite, and nitrite is converted to nitrate. Here the enzyme, the bacteria which are involved is Nitrosomonas and Nitrococcus. Here the bacteria which are involved is Nitrobacter. And these Nitrobacter, Nitrosomonas, Nitrococcus, they are chemoautotroph because they use inorganic substrate as the source of energy, which is nitrite and nitrate and ammonia. Remember this. And the bacteria which are involved in denitrification are Thiobacillus and Pseudomonas. Remember the name of the bacteria. A represent ammonification. Where the decomposition of the organic nitrogen of dead plants and animals, it's converted into ammonia, and the process is known as ammonification. This ammonia is released into the atmosphere, but some is taken by the bacteria which are present in the soil, and the rest is released into the atmosphere. And the bacteria which are involved in ammonification are Bacillus vulgaris, and second one is Bacillus ramosus. Remember the name of the bacteria which is involving in each step. It's important. Okay. Now look at the option. A is ammonification. The bacteria involved is Bacillus vulgaris. So A is, B is denitrification, and the bacteria involved is Thiobacillus and Pseudomonas. Thiobacillus. Okay, see students. Here they are given A and B separately. Okay. So here it's given A. A, we know it's ammonification, right? We know that bacteria involved in ammonification is Bacillus vulgaris, but Thiobacillus is involved in denitrification. So it's not. B says is Pseudomonas. B involves Pseudomonas, but not Nitrobacter. C says A is Bacillus mycoid and Bacillus ramosus. B says Nitrococcus and Nitrobacter. Let's keep it A and B, okay? This would for B, A, Bacillus vulgaris, and B, Thiobacillus for B. I think so. Remember that A denotes ammonification and B denotes denitrification, and the bacteria which are involved in ammonification, denitrification. Okay. Next one is choose the incorrect match. A is mitochondrial matrix Krebs cycle. Yes. B is F1, F not paired, which is oxidative phosphorylation. Yes, phosphorylation means ATP formation. And in mitochondria, in the inner membrane, there's oxidative phosphorylation occurs because when the electron are carried through the electron carriers which are present in the inner mitochondrial membrane, when the electrons are carried, the oxidation reduction reactions occurs, because of which the proton gradient get developed, and these proton gradients will play a very important role in ATP synthesis. C says the sodium potassium pump is active transport. Yes, because it requires ATP. D says aquaporins is the active transport of water. No, it does not require ATP. Active means involvement of ATP, but in aquaporins, no ATP is required. Aquaporins, they are the membrane proteins. They involve in facilitated diffusion. Why facilitated diffusion? Because number one is that they are the membrane protein. When the diffusion occurs through the membrane protein, the specific protein, then it is known as facilitated diffusion. And the movement is along the concentration gradient. That means it's moving, my molecules are moving from the high concentration to the low concentration. Choose the incorrect match. So D is the incorrect match. Okay, I hope it is clear to everyone. Thank you all for your participation. Thank you all, and see you in the next class.